Okay, everyone, let's pick up right where we left off. So we left off talking about different types of mutations, and now I want to continue on with microbial genetics and take a look at a couple concepts that are important here. We're going to look at genetic recombination and horizontal transfer and kind of answer the question, how do bacteria get diversity into their species? Remember, bacteria reproduce asexually through binary fission, so that's not going to create diversity in the offspring. But some of these processes can. Now, let's define our terms. Genetic recombination is the exchange of DNA between two DNA molecules. So a DNA molecule here and a DNA molecule here can exchange pieces of themselves with each other. This occurs within one organism and occurs naturally. It's known as crossing over during meiosis. Meiosis, the production of egg and sperm. It can also occur in the lab using all kinds of techniques known collectively as recombinant DNA technology. Horizontal transfer is the main way that bacteria can get diversity into their offspring, into new cells. Horizontal transfer is where genes are transmitted between organisms that are not parent and offspring. The two organisms can even be totally unrelated, and they can be different species. They don't even have to be the same species, although they could be. The transfer is between cells of the same generation, and this is important, horizontal transfer is not for reproductive purposes. Okay? It's just transferring DNA from one organism to another, or one cell to another, not for reproductive purposes. Now, this comes in several varieties. The first is transformation. Transformation is simply where free DNA is taken up by a cell and then integrated into the receiving cell's genome. Where does this free DNA come from? It can come from a living or dead donor cell. So, for example, here, let's say this cell up here dies. So A, B, C, and D represent genes on its chromosome here. All right? So this cell here is our recipient cell. Excuse me, this cell doesn't die. But this cell has A, B, C, and D. Now, somewhere over here, a different cell died, and its DNA leaked out. And its DNA was little a, little b, little c, and little d. And little a, little b, little c, and little d is just now in the environment. This recipient cell can take that DNA into itself, and then if you look here, it integrates it into its genome. Now notice that this cell now, down here, it's genetically different than what it started with, because it has these new genes that it uptook from the environment, from a dead or a living cell. All right? So that's very, very important here. This is showing it in a little bit more depth. I'm going to kind of skip over that. Transduction is another way that horizontal transfer can occur. This is where a virus transfers DNA into a bacterial cell. Remember viruses that infect bacteria? It's a bacteriophage. This can occur naturally, but it's used extensively in genetic engineering because we can tweak the virus so that the virus cannot cause infection. It doesn't release and destroy the bacterial cell, but it still injects its DNA into it, like this. So it attaches, entry, okay? Instead of forming new viruses and then bursting, right here, we can get this virus to transfer its DNA, that DNA, or whatever DNA we put in this virus in the laboratory, that DNA is transferred into the bacterial cell, integrated into its genome, and there's no release, no new viruses are made. So that's transduction. Conjugation is the third way. This is where plasmid DNA is replicated in a donor cell known as an F-positive cell, plasmid DNA now, and it's transferred to a recipient cell, F-negative, by direct cell-to-cell contact. Now, this transfer can occur through what's called a mating bridge or a sex pili, or pilus, singular. We see here. Remember in the chapter on prokaryotes, talked about this pilus here, or pili. It's a long strand used for transferring plasmid DNA from an F-positive cell, a cell with the plasmid, to an F-negative cell, a cell without the plasmid. Or it could be a mating bridge. Note these two cells are much closer together, and the cytoplasm here merges momentarily. So it's always plasmid DNA. So the main chromosome here is not used. So here's my F-positive cell. It has this plasmid. And remember, plasmids can give lots of advantages, so you want that. Here's the F-negative cell. It does not have the plasmid. They are connected by a pilus here. This F-positive cell makes a copy of its plasmid. It goes through the pilus into the F-negative cell, and then that pilus separates. Now, this cell also has the plasmid, and it has gained genetic diversity because it now has the genes that were on that plasmid. So this summarizes those three methods of horizontal gene transfer. Now, what's the significance here to evolution? The role of mutation, which we talked about in part 1, recombination, and horizontal transfer in evolution. Okay? All three processes, and this is what you want to remember, increase genetic diversity. Why is that important? Because diversity and the processes that create diversity are key driving factors in evolution. Bacteria have short generation times. They reproduce quickly. So these factors that increase diversity, combined with the short reproduction time, allow bacteria to adapt quickly to a changing environment. Only the most fit individuals have a better chance of surviving and reproducing. For example, a cell with a plasmid that gives it advantages might survive and then be able to reproduce better than a cell without those plasmid genes. The species survives and then better adapts to its environment, and this can continue indefinitely, especially when the environment changes. Okay? Now, bacterial genomics is the study of bacteria through their DNA, not through selective or differential media or those techniques we talked about in a previous chapter, but through their DNA. Over 180 genomes have been sequenced since 1995, the first one being this influenza virus. Not surprising that a virus was the first genome sequence, being that their genome is so small. Now, we need to remember that genes code for proteins, but they also code for other products. Proteins are made through the central dogma. DNA to messenger RNA to protein. That's transcription and translation. Genes can also code for something called transfer RNA. Okay, pictured here. Transfer RNA or tRNA, so DNA is going to make transfer RNA. So the gene in DNA makes a piece of transfer RNA. Transfer RNA is very, very important during translation because it carries the amino acids in and reads that genetic code, those codons. Ribosomal RNA is another product that genes can code for, rRNA, DNA to rRNA. Ribosomal RNA makes up the mass of most ribosomes. So a ribosome is really just a big ball of ribosomal RNA. Now, of importance, is a part of ribosomal RNA called 16S ribosomal RNA. This 16S rRNA is only found on bacterial ribosomes, and it's very highly conserved, meaning it has not changed much through evolution over time. So only bacteria have the 16S portion of their ribosomal RNA. Archaea don't have it, and eukaryotes don't have it, but archaea and eukaryotes have different structures in their ribosomal RNA, and that's what separates these organisms out into their three domains. Each is structured differently. All right? So there's that 16S. Bacterial up here has this 16S ribosomal RNA that makes up its ribosome. In a eukaryote, it's this 18S and 28S. So the ribosome is structured differently. All right, now finally, metagenomics is the study of microbial genomes, studying the genes of microbes from environmental samples. So the bacteria are identified by their ribosomal RNA sequences. All right, not by DNA, but their ribosomal RNA sequences. It's estimated that over 99% of bacteria are simply unculturable. You really can't grow them using those standard techniques we talked about in the chapter on growth and nutrition. However, almost all of them can be identified looking at their unique ribosomal RNA. So, for example, in the salivary microbiome, now this is just bacteria that would be in your saliva as part of your normal microbiota, your normal flora. Now, everyone, the size of the font indicates how common they are. So Streptococcus is really, really common. Then there's these little ones here I can't even read that are much less common. But I want to draw your attention to this right here. Look how big unknown is. These would not be culturable. They would have to be identified by perhaps DNA or rRNA sequences. All right? Okay, so that is it for the chapter on microbial genetics. Next time we're going to shift gears and shift from what microbes do to how we can control them. All right, so that the next chapter will be on physical control. All right, so that's it for today. Until next time, have a good rest of the day. Bye-bye.